Hello, everybody. This is going to be our Chapter 1 lecture for this course. Again, just a reminder that the lecture doesn't replace the reading. I will touch on some of the bigger main topics here, but there are a lot of things in your reading that still need to be covered, so make sure that you do read the chapters as well. So we'll start talking about the early years of microbiology, and so the text does a really good job at introducing Antony van Leeuwenhoek as one of the main protagonists of this first chapter, and well, he was a merchant, and he wasn't necessarily a scientist by trade, but one of the things that he did was he was concerned with the quality of the materials that he was using, and so he developed this device instead of using a magnifying glass to try to look at the fabrics a little bit closer up, and so that's kind of where we have the first powerful microscope that's able to look at things that are really, really small. And so later on, he was a very curious man, and so he decided to start looking at water. And so when he started looking at water from a water body, he realized that there were tiny things in there, and he describes them quite well. In fact, he describes most of the groups that we're going to talk about in this course when it comes to their taxonomic classification. And so he called them animalcules or beasties and things like that, as you will see in your reading. But essentially, what he did was he introduced the world to the world of microorganisms. And so here he is observing one of these little microscopic seeing devices. And here's kind of what they look like. So they were just a little piece of metal. It had a hole. He put a lens in there, and then he put the specimen up to that lens, and then he held it up to the light, and together they kind of produced some really good images. And I'm sure you can find some of those images on the web if you wish. So here we have some of the things that he saw. So here we have a couple of different things. We have some Daphnia, we have some filamentous algae. At the bottom over here we have some mites and some volvox, which are some other species of algae. So when it comes to the earlier years of microbiology, Leeuwenhoek was classifying things based on the taxonomic system of the time. And so this was around the time that Carolus Linnaeus was developing the system of organization for taxonomic groups of organisms. And so 6 categories he was describing, and those are bacteria, archaea, fungi, protozoans, algae, and other small multicellular animals, which we'll talk about later as well. So the first group, the bacteria and archaea, these are prokaryotic organisms. They don't have a nucleus. They tend to have something known as a nucleoid region. They have ribosomes throughout the cytoplasm of the cell, and they're very, very small, tiny, in fact, compared to a eukaryotic cell, which we'll look at in a minute. They're found just about everywhere. There's not a surface on this planet that doesn't have them. In fact, we're outnumbered by the number of cells of bacteria than we have of cells of our own kind. And so they're everywhere, from the most extreme environments to the most middle loving environments, and we'll talk about those as we go through the course. They also reproduce asexually, so they don't have a sexual means of reproduction, and we'll talk about why that's important in terms of their ability to accumulate mutations later on in the course as well. And their cell walls are mainly composed of peptidoglycan. Some of them don't have some of these cell walls, but overall they tend to have some sort of protective barrier around them. And then we have archaea cells, which are a little bit different in their composition. They're a little bit more closely related taxonomically to animal cells than they are to bacteria, at least in some aspects of their metabolism. So here we have a human cell. I think these are buccal cells, so cells from your cheek, and some bacterial cells. So this is something that you can get on a basic light microscope. And if you treat these cells with methylene blue, you could actually see some bacterial cells around. And you can see they're tiny, just very, very tiny, compared even to the nucleus of these cells. So how can we look at organisms and find something meaningful? Well, there are different processes, both industrial, commercial, things that we do even in our homes that are quite important, and some of them including, and historically speaking, your text does describe a lot of these things like Saccharomyces cerevisiae and Candida albicans, which are fungi of very, very high importance, as one of them is really important for production of food, bread, cheese, that kind of stuff, and the other for disease in humans. And so they're both pretty significant. And so again, just a reminder to make sure that you are reading the text. But in this case, these are eukaryotic. So now, in contrast to prokaryotic organisms, these have a nucleus. They're typically external digesters, and then they'll absorb those things. They have cell walls, and these cell walls are very, very different compared to anything else. In terms of molds and yeasts, these are the two subclassifications of fungi that we're going to be dealing with. Typically, the molds are going to be multicellular, so you're going to have filaments, things known as hyphae, that can generate spores, and yeasts, in contrast, are unicellular. So they're going to be predominantly spores that reproduce, and these can produce both sexually or reproduce sexually and asexually as well. So here we have an example of these. So on the left you have the canidia, which are the reproductive structures of some members of the genus Penicillium, and on the right you have yeast cells, which are, as you can see right there, they're budding, and so budding is the asexual reproductive cycle of these fungi. Now protozoans are protists, they belong to that group. They're single-cell eukaryotic cells. They're really similar to animals in both structures and metabolism. They usually like to live in aquatic environments or semi-aquatic environments. Some of them are vector-borne, so they live within other organisms, and that's how they reproduce. So they are asexual, but some of them have sexual reproductive capabilities. And then you have different mechanisms of locomotion for these, and I'll show you an image of what those look like in a bit. But they could have pseudopods, which are false legs, things like amoeboid motion. That's the stuff that we're talking about. Cilia. Cilia are tiny little hairs that kind of flick back and forth. And these cilia are not very strong. They can orient the organism in a specific direction. So as the current of water moves, they'll move with the current, but they'll guide themselves to where they want to go. And then we have the flagella, which are the whip-like appendages that these can swim inside the current and slightly against it. So on top we have our pseudopods, and then we have our little cilia in the middle, and then we have the flagella in these that appear to be euglenoids, which we'll talk about a little bit later. So then we have algae. So algae are also protists. They're unicellular or multicellular. They're photosynthetic. Oh, and I forgot to mention that some of these protozoans can be both autotrophic and heterotrophic, so some of them can consume or hunt and photosynthesize at the same time. Algae, predominantly, they're photosynthetic. They have very, really simple reproductive structures, and we'll talk about those when we get to that section. But some of them are going to be characterized mainly by their pigmentation and the composition of their cell wall. So here we have two examples of them. On the left, you have filamentous algae, and on the right you have some diatoms. So diatoms are one of the most diverse species of algae in the world. So they're everywhere in every body of water. They get dispersed throughout the world via wind currents, and so you'll find them in the oceans, you'll find them in lakes, ponds, streams, estuaries, everywhere where there's water, you will find diatoms. So there are others that we study, parasites and viruses. Now, these were a little bit different than the others. So parasites include worms. So worms are something that we still study because they are unable to be seen without the aid of a microscope. And viruses, of course, we kind of predicted their existence a long time ago, but it wasn't until the discovery of the scanning electron microscope that we were able to visualize them because they were so small. Here we have some of those parasitic worms parasitizing human blood. And here we have bacteriophages. So there are different types of viruses. I know these are very popularized by the media as, very infectious and things like that. These will only infect bacterial cells. We'll talk about the other types of viruses that infect everything else, right? But again, we knew there were smaller things. Initially, they called them filtrable viruses. Again, it wasn't until like the 1930s that they were able to see them. But after that, they were pretty Pretty interesting. We found things like yellow fever in humans caused by some of these viruses. And of course, now we have the entire branch of virology. And then getting to the golden age of microbiology. So this is kind of where there was a lot of advancement in this field. And there were four questions that were responsible for this. So there were some interesting scientists that we'll talk about, and you should know some of their important contributions. But the questions are, is spontaneous generation of microbial life possible? So can we get life from abiotic places, environments? What causes fermentation? What causes disease, and how can we prevent diseases and infections? And so some of the earliest scientists, they were perplexed by some things that they saw, and they knew that there were processes that gave rise to life, and one of them was asexual reproduction, cloning of organisms, budding of organisms. Sexual reproduction they knew as well, quite well, but then the abiotic origin, or the non-living matter, these were ideas that were popularized by philosophers like Aristotle a long, long time ago. And so when we're thinking of these ideas, just remember that for a long time, people had these notions and preconceptions of how things worked and how living things worked. And so it wasn't until the microscope and some of the people that came after some of these important figures that we really understood how these things worked. Um, and so in order to disprove that spontaneous generation, everybody was kind of like, they were in a different place. Some people agreed with it, some of them did not agree with it so much. And so one of the first people that was looking at that was Reddy. And so his experiment consisted of allowing decaying meat to be out in the open. And so when he left this piece of meat out in the open, he noticed that after some time, maggots began to form, and then insects came and everything else after that. But when he covered this meat and wasn't exposing it to the outside environment, he realized that there were no flies. And so they began to question what Aristotle's ideas or views of how living things originated. And of course, this was already happening. I mean, in the 1800s, mid 1800s, there were people like Charles Darwin Jr. that was publishing things like On the Origin of Species that was already changing what preconceptions people had about life and change and things like that. So these views that Aristotle had were now starting to be replaced by new mentalities. And so here we have that example of Reddy's experiments where he had the open, the sealed, and then the cheesecloth or the gauze that, it attracted the flies, but the flies were laying their eggs and their larvae on the cheesecloth and not the meat. And so this was kind of interesting to a lot of people. And so other people came afterwards, of course. Needham was one of the next scientists that was researching or trying to develop experiments to say, well, yeah, large animals, they can't come from abiotic factors. There has to be something there. But he said that, well, because these other organisms are so simple and not complex like microbes are, they could. And so what he did was he took this beef gravy and he mashed up a bunch of plants and stuff like that. He boiled it for a long time and then cooled it, and he left it out, and he realized that, oh, you know what, things are growing here because it's getting cloudy, and the cloudy indicates that there are microbes present. And so he thought, or he came up with this idea of the life force, that as long as there are abiotic resources and oxygen, basically the things that are required for life, that the life force would cause the spontaneous generation of these microbes, and that's why things would spoil. And I mean, people were like, "Oh, yeah, that makes total sense." Except, that's the thing about being a scientist, is you always have to be a skeptic. And Spallanzani's experiments were done as that, as a skeptic, because he didn't believe that Needham's findings were accurate. He said, "Well, you know what? Maybe you're wrong," and he just developed this different experiment. So what we did for his experiment is really, really similar, except instead of leaving things open to the environment, he took these glass ampules and he sealed them after boiling them, and then he realized or he noticed that there was nothing growing in them. And so, haters gonna hate. And they were all like, no, well, you know what? You must have heated them too hot and you destroyed the life source, or things like, you sealed the vial so there's not enough air for things to develop naturally. And so, the people were just remember, a part of the scientific community is to be a skeptic. And I'm not saying we should question things that have been proven and are well documented, but things like this are really, really important, especially for establishing the stage for the people that come after us. Right? And so that's kind of exactly what happened later. Louis Pasteur was one of the ones that is credited as being one of the modern fathers of microbiology, because of the way his experiments kind of really, and his contributions as well, not just his experiments, how they changed the view of microbes. And so what he did is he set up a combination of these things, because he said, okay, well, yeah, we need to boil things to sterilize them. And okay, fine, you don't think that, maybe you think that air is really, really important, so let's provide some air to these flasks. And time, you have to give him some time. And okay, and so he was pretty convinced that he was on the right path. And so what he did was he set up these flasks that had a swan neck on them. And the swan neck allowed air to flow. But after everything was, killed off, and air was cooling this vial, now there were little particles of dust settling at the base of the neck. And that neck was essentially accumulating anything that was trying to go into these flasks. And so even though there was stuff accumulating there at the base of the neck, there was nothing making its way to the inside of the flask, and this kept these flasks from growing anything or from spoiling. And so now he didn't just stop there. He decided that he wanted to revisit that hypothesis. And so he said, well, if I tilt this flask and I introduce all this dust that's been accumulating into the flask, will it grow microbes? And so that's what he did. And what ended up happening was what he had thought all along, is that the microbes, or these organisms that were causing things to spoil and that were growing, they had to come from somewhere. And that dust was the place where they come from. He couldn't say, or anybody couldn't say, oh, well, maybe you heated it up and it had lost life force, or they're not getting enough oxygen. Now there was something that was directly cause and effect. If you keep microbes out, they will not spoil, but the moment you introduce them to the external environment, that's when you start having spoilage, because organisms are found out there and not in a sterile environment. And this is known as the process of pasteurization, where we heat things to a certain temperature to kill everything off, and then we seal them in different ways so that it prevents the spoilage of our products. And so we do it to things like milk. We do it to just about everything that you get at the grocery store. It probably has been pasteurized, and it'll say so at the bottle, or at some place on the bottle or the container. So again, there has to be some sort of experimentation in order to prove these claims or make statements regarding these claims. I don't want to say necessarily prove, right? But the scientific method is really, really paramount for understanding how these things work. And so there was a debate, of course, of something happening a certain way. That question or the statement was what led to developing a hypothesis. Then after that hypothesis was developed, experiments were developed to test these hypotheses. And of course, those hypotheses have to be testable. And once they were testable, they were either supported or disproven. And so you either accept this hypothesis, and by collecting further evidence becomes a theory or a law, or if you disprove it, you can go back and revisit your hypothesis, or just say, this is not the case. And we do need that. Just think about a medication. If we were to look at different types of medications and we want to say, is this effective for treating this disease? And we end up concluding that it doesn't, we shouldn't modify that hypothesis and just say, this is ineffective at treating this disease, condition, disorder, whatever it is. That's why we need this. And if it worked every time, then it wouldn't be science. It would just be validation, right? And so that's a really, really important part of the scientific method. And so here we just have a graphic kind of showing us the way this would proceed. So then the next question that was asked was, what causes fermentation? And so some people knew that um, if you left these things in their natural environment, sometimes they would spoil. And for example, making wine, they knew that if they crushed the grapes and then they placed them into bottles, eventually they would form wine. But if they tried doing this long term, these would spoil. And so they were curious as to why some of these things would spoil, but then others wouldn't. Like this was not something that they had issues with things like beer, for example. And so the debate over what causes fermentation sparked another, set of experiments. And there we have our friend Pasteur coming into the picture, saving the day. And he said, well, having air in some cases is what causes fermentation. And so he did a series of experiments, and then he discovered that, well, there are these organisms that are called facultative anaerobes that can choose what type of metabolism they're doing. They're either doing or performing their metabolic activities in the presence of oxygen, or they're doing these in the presence of no air. And depending on what conditions you have, what organisms you have, they lead to different things. And so one of the things that he noted was that, well, bacteria tend to do something different than fungi. And so part of the process of pasteurization initially was heating up the flask and the liquid or the fluid, whatever it was, to a specific temperature to kill whatever was there. And so specifically, what this did is it killed bacteria. And the bacteria that was dead wouldn't spoil the wine in this case. So it wasn't after you boiled it that you introduced the yeast, and then this yeast was responsible for fermenting into alcohol, or the grape juice into alcohols. And so then we know, of course, now that there are different organisms that have different products. In the case of bacteria, we know that they can ferment things and create something called lactic acid, which will eventually spoil things like your food. Whereas fungi, when it ferments something, it'll create alcohols. And so now we know that we can actually do that, and we do it every day. In industrial places like the industry for making cheese and wine and even bread, fermenting chocolate, all these things. We use these organisms because we know what they do, and all these were because of experiments that were done years ago. And so again, pasteurization was or is a very, very important process that we use until today. And we're probably going to continue to use this for a very long time. And now we have the field of industrial microbiology, where now we can use some of these microbes for manufacturing products for our use and consumption. So cheese, alcoholic beverages, soy sauce, vinegar, yogurt, all these different products, they require bacterial and fungal cultures of organisms in order to be produced. Not only that, but we can get things like antibiotics from both bacteria and fungi. We can get hormones by modifying bacteria genetically to produce them for us. So there's a lot of different things that we have applications now because of these experiments from long ago. So then Buckner came along and he argued and he said, no, I disagree with the idea that you require living organisms to do this. And so what he did is he showed that enzymes would actually be able to do these processes without the living organism. And so that is kind of where we have the field of biochemistry coming in, because yes, technically you can use the enzymes to do those things for you without actually having the microorganism. So then Pasteur started developing the germ theory of disease, and it wasn't until another scientist a little bit later on that we actually solidified something, and that's kind of where we introduced Robert Koch. So Robert Koch was a scientist that worked specifically with anthrax, and so one of the things that he did was he was trying to find a cause or a relationship between some microbes and diseases. So what causes that disease? And so what he did was he looked at these microorganisms, and every time that he would take a culture from something that had anthrax, and he would inject it into like healthy mice, he found that they showed some of the same symptoms of the sick animal he took them from. Now, of course, we have ethical considerations, of course, but we still do a lot of animal testing and things like that in order to understand how some of these works. And we have other models as well. Not all of them are vertebrate animals. And so one of the things that we have now are all these techniques that help us visualize these microorganisms, such as the photomicrograph of bacteria. And we now have things like autoclaving and the use of culture plates for medium and everything like that because of these advances. And Fanny Hesse was a very important scientist as well. So she worked with her husband in Koch's lab. And one of the things that Koch was trying to do was just grow these things. He was trying to create cultures of bacteria where he could take them and experiment on them. And he realized that, well, he would take potatoes, slices of potatoes, and then he'd try to inoculate them with his bacteria. But he realized that a lot of different species of bacteria would eat the potatoes. And so he would not be able to differentiate between one type of bacteria from another. So when Fanny Hesse came along and proposed the idea of using this thing called agar, now finally he was able to make some progress. So that's gonna allow for the, I guess, for the separation of these cultures. And so each one of those species of bacteria that were present in his samples, they were creating discrete colonies, as you can see here. And so each one of those spots is a colony of bacteria, and they're consisting of the progeny of that single bacterial cell that was captured by one of his swabs. And so, of course, now from there, you can isolate things, you can grow them in another plate, and then essentially create this plate that only has one species of bacteria. And so he developed these postulates to help link the disease to the microorganism. So he called them causative agents. He didn't want to call them anything else because he believed that they might cause that disease, and so that's why the term causative agent, because it could be a bacteria, a fungus, a parasite, something else. So they have to be present in every individual that's sick and absent in every individual that is healthy. So you have to be able to extract it from that sick individual or organism, grow it outside of the host, and then culture it outside of the host. Then you have to introduce it into a healthy organism that's susceptible, and they have to show the same symptoms of that disease. And then you have to be able to take it out of that organism again. So you have to be able to take it from the sick individual, culture it, give it to a healthy one, and then pull it back out of that healthy one. And so this kind of establishes the link between the microorganism and the disease. And so it's a very, very important in the germ theory of disease. So here we have a list of a bunch of scientists, the disease they worked on, and the agent that caused that disease based on the establishment of these postulates. So make sure that you take a look at that. So now when it comes to visualizing these organisms, this was something that was also done in Koch's lab, and that was the Gram stain. It's a very, very useful stain. It's one of the most widely used, and it's kind of one of the first things you do if you want to start identifying bacteria. So it's really useful because it can determine the properties of the bacterium and then allow us to treat it based on the type of bacteria that we have, the size, shape. It allows us to classify them. And what it does is it uses a stain and a counter stain to differentiate those cells that are rich in things like peptidoglycan, or those that don't have it, or if it has a double or a single membrane. Typically, it starts with a crystal violet stain, and then you add gram iodine as a mordant to make it kind of stick to the inside of the cell. Then with a decolorizer, you remove any stain that didn't stick, and you could use things like acetone or ethanol to remove any excess, of course, and then you introduce the counter stain. In most cases, that counter stain is safranin. And so what you'll have is you'll have images that kind of look like this, where you'll have bacterial cells that are stained purple and some that are stained pink. The ones that are purple, those are gram-positive. So when you think of gram-positive, the P is for purple, and gram-negative, the N is for the N in pink. So just a way to hopefully help you remember them. But as you can see, those purple ones, they have the crystal violet, and it's inside that cell, and it's stuck there. It didn't leave. And all the cells that were stained pink, those are the ones that didn't accept the dye or didn't take that dye. And so when they were rinsed with the decolorizer, all the crystal violet was removed. And with the counter stain of safranin, they're actually going to look pink. And this is one of the things that allows us to visualize the differences between the composition of the cell wall of these bacteria. So now when it comes to how we can prevent diseases and infections, there were a lot of different scientists that contributed to this. But some of the more notable ones, or that ones that are discussed by your text, are Semmelweis. And so his hand washing processes were very heavily criticized, and you have to feel sorry for the guy at some point because of the way he was treated compared to the contributions that he made to the field. And so he was one of the earlier ones. He believed that whenever medical students were learning and dissecting human cadavers or were doing their autopsies, they carried with them something known as the cadaver particles. And these cadaver particles were some that led to a type of fever in patients that were delivering babies. And so when the babies were delivered, sometimes these cadaver particles that were there would cause them to die, have a fever and die. Now we know that it's actually a type of streptococcus that causes that, right? Like, it's typically not that bad on your skin, but once it gets into an open wound or your blood, then it becomes really, really terrible, right? And so what he did was he asked his students to wash their hands with chlorinated lime water. And they used this in these institutions to kind of clean the rooms so that, like, if a cadaver was very, very smelly, that odor of the cadaver would disappear. Of course, and this was, they just thought he was crazy. They made fun of him, so much so that the manager of the place where he practiced, he got rid of him. He was like, nope, get out of there. And he tried convincing a lot of other practicing professionals to do the same, but no, he was just made fun of, and the poor guy went crazy. He was eventually committed to a mental institution. And I don't know, it's just like the worst thing is he died of a streptococcus infection, which was like the thing that he had worked to eradicate throughout his career. It's really, really sad, but it's not all bad. After that, Lister came along, and with his antiseptic technique, he was an interesting guy as well. He kind of took some of that information from his predecessor and said, well, if you clean these things, it might be a reason why they survive. And so he didn't use chlorinated lime salt like Semmelweis did. He actually used something known as carbolic acid or phenol. And so this was another thing that they used to kind of deodorize things, but this one was in the sewage. And so what this did is whenever he would spray it on the people that he was that I guess they were working on, it led to a decrease in their death. It was pretty significant too. And so once he convinced people that, hey, you know what, less people are dying when we spray the wounds or incisions with carbolic acid, like, they don't die, they survive. Finally, they, accepted that idea, and he did his best to try to, put in a good word for his predecessor so that he wouldn't be remembered for being ridiculed, but as one of the people that established these ideas of cleanliness and sanitation in these environments. And then, of course, we have Nightingale. So the main cause of death during war wasn't always the enemy. In fact, some people say that the diseases that outbreak or that they break out during war are what wins them. And so Nurse Nightingale was one of the ones that helped um, push for the practices of sanitation in these conditions. She said, you know what, you shouldn't be changing your dressings from war in the same place where you came in. And then the environments that they were using, they were to be scrubbed and cleaned by any of the staff that was working. And so after, saying things like, you know what, the conditions here are terrible, the food is terrible, people are dying not because of the war, but they're dying because of disease. And so she took this later on to England, where she established these practices of cleanliness in the field of nursing, and it's led to tremendous success. These things are still practiced today. The cleaning and sanitation are extremely important. And of course, now we know that there are different types of pathogen. We have our soil-borne pathogens, our waterborne pathogens, and now we know that there are also blood-borne pathogens. And so we do the best we can to minimize infection by following some of these practices still today. So then, other important figures when it came to preventing infection of disease. Uh, Snow was a very, very important figure for infection control. So there were a lot of issues in Europe, and I'll talk about why disease was so important. Is because Europeans were just very, very bad at dealing with their wastewater. Um, it sometimes mixed their black water, which contains fecal matter and urine, with drinking water or potable water. And so what happened was that people were drinking this dirty water and they were dying of cholera, right? And so one of the things that he used chemicals to try to prevent the emergence of these diseases, kind of to stop the disease of cholera, because people were just dying. Like doctors would just visit patients, they would look at the symptoms and say, oh yeah, you're definitely sick. There was really nothing they could do at the time. And so by preventing the spread of cholera in the drinking water, it was one of the things that led to the survival of a lot of people. So treating it at the source essentially was what saved people. So Snow was one of the people that kind of started that idea. And then we have Jenner's vaccine. So one of the things that he's credited for is for variolation, which is highly regarded by many as a bad move. But it was a really important start in the field of immunology. And so here we have Lady Mary Wortley Montagu, who had been to probably the Ottoman Empire, I believe, where some people had reported that if you took like little fluids from the blisters of people that were diseased, not necessarily critically diseased where they were going to die, but very mild cases of disease, if you took those fluids or that discharge and then you inoculated a healthy patient with it, they would get sick, but be immune to the more strong or more virulent form of the disease. And so it was effective to a degree. The bad part was that they didn't consider that there were people that might be immunocompromised or have different conditions. And so in some people, the disease was fatal. So they tried protecting them, but they ended up killing them. And so there's a lot of different characters here. But of course, this had been practiced different places around the world, probably since 200 BCE, where people discovered that being close to other people that were sick might have caused the disease, but also made you less susceptible to the same disease in the future. And there were a lot of observations that people would do, like milkmaids, when they were around cows that had cowspox, they would be exposed to this virus, but they really wouldn't get too sick. The symptoms were actually fairly mild. And so when they were exposed to smallpox, they were pretty much immune to it. And so other figures like Jenner, who came after Lady Mary Wortley Montagu, um, and Louis Pasteur, they developed what we now know as modern vaccines, right? And the word vaccine derives from cow. Like if you're a Spanish speaker, vaca, or cow, that's kind of where the origin of that word is. So vaccinia virus is um, kind of saying where the virus came from the cow. But yeah, of course, there are many other people that are fairly notable, so make sure that you do read that section in the text. There are a few slides that talk about the fields of microbiology. Um, I do expect you to know the uh, basics of them, so what each one of them is centered around. So now the question, what are the basic chemical reactions of life? And when we start looking at biochemistry, we have to consider Pasteur's work some of the most critical. Um, Understanding the role of the genes and the proteins and the metabolic processes that are involved with some of these organisms is really important to understanding how things really work and why we use specific model organisms for specific reactions. And they have many different implications. Anything from designing herbicides and pesticides, treating illnesses, treatment of diseases, designing drugs and manufacturing them. All these things are very important. And so that kind of leads the way into the modern age of microbiology. Um, so microbial genetics predominantly deals with how we take genetic information and we translate it into a protein that's functional that does a specific task. And so we can keep track of mutations, the mechanisms by which things mutate, and identify anything that we can use to try to modify that expression of genes. Um, Again, we can take the sequences and we can say, well, this sequence leads to this protein and this protein does this. And so we know now the cause and effect of those genes. We also are able to look at the sequences and establish relationships between different taxonomic groups. And so one of the things that this allows us to do is to do different types of sequencing. So we could do environmental sequencing where we can take just whatever's out in the environment, grind up those fragments of genetic material and look for genetic markers that are specific for some species of bacteria. So there are some types of bacteria that are really difficult to culture. An example of that is the cat scratch disease, which is caused by it says an unculturable organism in your text. It's actually Bartonella hensley, and it's a really difficult bacteria to grow in culture. But if you're using serology, which is the study of some of the fluids and serums that are circulating through your system, and those that are found in wounds, when you couple that with PCR, you can actually find that there are genetic differences to that specific bacteria compared to any other ones that are found on the skin. And that's one of the ways that we know that they exist. It's not necessarily because we've cultured them, but because we know that the sequences are there. Now, when looking at DNA technology, we have now the power to recombine DNA. That is to take a fragment of one organism and introduce it into another. And the reason we can do this is because of the universality of the genetic code. So hopefully you remember from general biology that all living organisms that are made of cells and have DNA have to have adenine, thymine, cytosine, and guanine, the A, T, C's, and G's, or the code of life. So if you're an organism and you have a specific code, that code can be transcribed and translated by cell machinery, like if it belonged to you. And so we can take, for example, a toxin that's produced by a bacterium and introduce it into corn. And if you put it in corn, now corn can make that toxin that the bacteria that you took it from makes. And so I know it seems kind of like, well, why would you want to do that? Well, we can do that because let's say that we're trying to protect this crop from a lot of insects that are causing millions of dollars of damage. And if you give corn the ability to make its own pesticide, that could save millions of dollars, right? And so we don't look at it that way all the time in terms of money, there are things that we can do, like producing clotting factors in E. coli by giving the gene to the bacterium, and fixing genes by introducing functional genes and replacing the non-functional genes um, in different cell lines. Now, in the environment, we have different uses for these things. There's the idea of bioremediation, or the cleaning up of environments using bacteria, fungi, algae, and we could do this two different ways. There's typically seeding and fertilization. Seeding is where you introduce an agent that isn't there before, and then it grows and it cleans up your polluted area. Or you can do fertilization, which proliferates the microorganisms so that they grow or they bloom, and then they'll take care of that contaminant for you as well. So what do we do to defend against diseases? Again, serology is the study of the fluids, or the serum in the blood. And what we can do is we can look at it and, okay, we know that there are things within it that can fight infection. And immunology is just the field that looks at the body's defenses in response to pathogens. And we will study a little bit of immunology in this course in some of the later chapters. And then, of course, chemotherapy. I know I skipped one scientist, I believe, while looking at the magic bullet. Well, we know that there are different chemicals that kill different types of bacteria. One of the earliest ones was arsenic. I think it was Eldrick that was doing that. But of course, we know arsenic is also dangerous to humans, but that was kind of the start. We know that there are things that can kill organisms and only kill those, and we know, or we call that selective toxicity. And so there's different drugs like penicillin and sulfa drugs that can kill some species of bacteria, but essentially be harmless to others. And fungi are really interesting. Fungi are kind of where a lot of antibiotics come from, or came from at some point in time. Uh, so fungi will release different chemicals that are antibacterial, so they'll prevent bacteria from growing on them. So here you can see something called the zone of inhibition, and we will actually get to practice this in one of our lab exercises. So you'll have an area where you have fungus, and then you'll have a place where nothing grows, and then after that distance, then you start seeing the appearance of bacteria. So this fungus is repelling the growth of bacteria within that small region. Now, what will the future hold? Well, there's a lot of questions. The more questions we answer, the more questions we have. We are very, very curious, and we always ask why and how. And those whys and hows have got us to where we are today with our current existing knowledge of these microbiological processes. And so it's important to ask questions, answer questions, and ask how and why things work. And just like every scientist that we looked at, a lot of them were skeptics. They were asking questions like, well, does this work? Why does this work? Why does this not work? And so that is what led to a lot of different discoveries. And so it's also really, really important to be a skeptic on top of this. Okay, but that is the end of this lecture.